Welcome you, my dear students. We are watching a series of video lecture on RNA processing. RNA processing, before going to understand the RNA processing, we need to know the types of RNA. And already in the previous lecture, I have discussed about the types of RNA and their functions. Okay. So today, in this video lecture tutorial, we are going to start from ribosomal RNA. Okay. So here is the structure. You can see this is the 3D structure of ribosomal is the small subunit and large subunit, as we all have studied during our 10th standard or 11th standard. It's like the cup plate, as for example. So this is a large subunit and small subunit. They just attach between each other is the ribosome, and ribosomes are the sites of protein synthesis. All of you know that only in the ribosome only protein can be synthesized. Okay. So here in the picture you can see the tRNA molecule attached here growing a polypeptide chain is the mRNA, and here is the small subunit, this one, this is the large subunit, and in between tRNA just enters here. tRNA, they just read the code of mRNA triplet codon. As per the codon, the amino acid will be synthesized here and one by one chain will be formed. Okay. Well, about the ribosomes, they consist of ribosomal RNA 65% and proteins 35%. Okay, this is a very important and informative data. Ribosomal RNA, actually they consist of 65% in the ribosomes and protein 35%. They have two subunits, that is the one is large and small subunit. Now we are talking about the transfer RNA. Transfer RNA is the smallest RNA in the RNA types. The existence of transfer RNA was demonstrated by Hoagland in 1957. You can remember it because this is important for MCQ. And anticodon loop has three nucleotides that function as anticodon. The anticodon loop has three nucleotides. We know that the codon and anticodon. That means codon is in the mRNA and anticodon should be on the tRNA. So anticodon should be matches with the codon. When anticodon from tRNA matches with the codon on the mRNA, then amino acid synthesis will be started. Okay. So thymine loop function as the ribosome attached region is not so important. And base pairing involve hydrogen bond. This is a very important point. Base pairing involve hydrogen bond. You know that during codon and anticodon base pairing, in between only hydrogen bonds will be there. Is the normal tRNA three-dimensional structure? That means tertiary structure of L-shaped tertiary structure, because 1D means one dimensional is the primary structure, is the linear structure. 2D two dimensional is the secondary structure, but 3D is the tertiary, and 4D quaternary, or so on. Now is the messenger RNA. So messenger RNA is who carries the message. Okay, message for amino acid code. It is a single-stranded base for base complementary copy of the DNA strand and provide information for amino acid sequence of a polypeptide. So who carries the information of amino acids? This is the messenger RNA. The message carrier is messenger RNA. Carries genetic information means the message from nucleus to cytoplasm. So nucleus, because DNA to RNA is the nucleus activities, then nucleus to cytoplasm means protein synthesis will be occur in the ribosome, so it will be transferred into nucleus to cytoplasm. Okay, so present in two forms. The messenger RNA is active support translation and inactive form means do not support translation. So mRNA basically present in the cell in two form. They are active. When they are active, they means they directly involved in the translation process, and when they are inactive, they do not participate in the translation process. This is a very, very basic thing. So here you can see the mRNA main three parts of a mRNA molecule. mRNA, at you can see at the 3' untranslated trailer sequence. This part is not translated, means not coded for any amino acid. This is the in between protein coding sequence, means all the three triplet codons are responsible for each amino acid. And 5' untranslated leader sequence is the trailer sequence, means the backward, and leader mean who lead from this one, this side it will be go on. So is the leader sequence, this portion of mRNA sequence, they lead the translation process, but they do not translate it because only non-coding regions are there at the 5' at 3'. Okay, only in between protein coding region is there. So in between here you can see is the translation start, means the initiation codon will be there, and here is the stop codon. So initiation and stop codon, they do not response for any kind of amino acid. Don't forget. Okay. So three main parts is already discussed, and is a very important thing, ribosome, because we are discussing about the protein synthesis and RNA processing. So it will be very understandable to know about the ribosome structure and the functions and what is the thing in actually the cellular organelle inside the cell. So these are catalytic RNAs that mainly participate in the cleavage of RNA. Okay, in the cleavage of RNA, because ribosome actively participate in protein synthesis. They cleave the RNA molecule. They are not true catalysts because they alter their own structure as result of catalysis. So once they are not true catalysts because they alter whenever required as per the termination codon, start codon, untranslated region, protein coding region. They actually alter their structure and results of catalysis. So for example, RNase P is the common ribosome that matures tRNA that act as an endonuclease. Endonuclease means which helps to cleave the RNA molecule, and self-splicing introns. Introns means those are non-coding regions. Okay. So now the clinical applications of RNA used in therapeutic agent in correcting mutant mRNA in human cells and inhibiting unwanted gene expression is very important. Is actually inhibit the unwanted gene expression, kill cancer cell. Okay, prevent virus replication, gene inhibitors. So here also is the repeated gene amenders. Okay, it can also amend the gene regulation, protein inhibitor, protein synthesis and protein inhibitor, immunostimulatory RNA. So these are the clinical applications of RNA rather than only protein synthesis. Okay, so these are very important point. You can note it down. So now come to the main slide, the broad topic of what is actually RNA processing. So if I talk about the food processing, if I talk about the rice processing, if I talk about the noodles processing, so how we will prepare. So this is similarly the concept, what is RNA processing, how RNA process and how RNA process to work to be more functional. So here are some definition or conceptual definition. Most newly transcribed RNA molecule, primary transcript is called. These are the primary transcript. Most newly transcribed RNA molecule from DNA molecule to RNA molecule. These are the primary transcript undergo various alteration to yield the mature product. That means once DNA to RNA is formed via transcription. So newly transcribed RNA molecules, these are called the primary transcript. they actually alters themselves to yield the mature product. Okay? Or the definition, RNA processing is the collective term used to describe the molecular event allowing the primary transcript to become the mature RNA. It's the same thing. So that means the collective term RNA processing means once the primary RNAs, means the primary transcript from is these are the very nursery level RNA is immature RNA. Okay, so because just they have transcribed from the DNA molecule, they are not the mature. So they collectively they will be altered themselves to become a more mature and stable RNA molecule. Once it is not a stable RNA molecule, it will not participate in the translation process. Or you can describe here the process of modification. So modification needed because the primary transcript are the unstable or immature, mainly through cleavage or splicing or cut the primary RNA transcript so as to release functional RNA molecule from them. So that means from DNA to RNA when is processing. So DNA to RNA, there will be some non-coding portion, there will be some introns. So you need to, that means these are the immature DNA, RNA molecule, primary transcript. So you need to cut, cleavage or splice the unwanted DNA, RNA sequences, okay, from the primary transcript so as it will act as to release the functional RNA molecule from them. Okay, so it carries out by the nucleases RNAs that cleave RNA. So this is a very important for MCQ only. That means it is nothing. Just you remember, DNA to RNA is forming. Once DNA to RNA is forming, so there the RNAs molecules are called the primary transcript. They are the immature unstable RNA because there are lots of unwanted bases are there. So once you cut using the RNAs, ribonuclease, and spliced them to become more stable and mature mRNA molecule. So this is all over the RNA processing. So I hope you understand the thing. So rest of the part we will continue in the next video. Okay. Thank you for watching.